I'm going to start off with the absolute basics, to be honest, which is probably not the bit that worries you, but nonetheless, I'd rather go through all of it from the start. So here we go. So just listen to me very carefully, folks. And then as ever, there will be some questions. Okay. So DNA is made of nucleotides. Okay. And we have a phosphate, a ribose or a deoxyribose sugar, and a base. Okay, a nitrogenous base. And obviously it's the nitrogen that means it's a base. But they're both hexos, as you'll see in a minute, and they connect with a condensation reaction. So this bond here between phosphate and sugar is called a phosphodiester bond. And it's a condensation reaction, so water will be expelled in that, as seems to be the fashion with these things. Now, those of you who are chemists could also think of that as a covalent bond. Okay. So this one here, we're actually going to call that, this might surprise you, I'm going to call that a glycosidic bond. Why would that surprise you? Because you say, well, hold on, sir, that's between two sugars, normally. Again, it's condensation. H2O is going to be liberated. I don't really have a reason as to why, although the base is very sugar-like when you look at it. But we're calling it a base because of the nitrogen involved, and the nitrogen is the big difference between the base and the sugar. And what's not shown on here is further bonding would take place here, which would be H bonding. Now the reality is that's going to be covalent, that's going to be covalent, okay? But we start naming them specifically. Okay, so, yeah, that's what I just said. Didn't realize it was coming up. So we'll go into more detail about the differences between RNA and DNA in a minute, but there's a ribose sugar and a deoxyribose sugar, and if you spot the difference with it, you can see deoxy, so it's been deoxygenated there as compared with this point there. Everything else is the same, and we can, just like with the lesson on carbs the other day, we can number the carbon atoms. So this is basic GCSE stuff. Okay, A nitrogenous base is always joined with T, and the way I remember that is straight letters like to get together with straight letters, and bent letters love to get together with bent letters. Okay, that just helps me remember it. It's going to come unstuck when we talk about A getting together with U, which we will do later on, because U is a more bent, sort of curvy letter. So that's where my theory of straight with straight breaks down. But of course, that's just a memory trick. Why is it actually biochemically only A joining with T? Okay. Oh, by the way, this at the bottom is sometimes there's a question on proportion, a bit of numeracy, and you can work out proportions, because if you know the proportion of C, then you will know the proportion of G, and then you will therefore know A and T. So if you know the proportion of any one of these four letters, you can work out the proportions of all the other letters. We are talking about DNA at the moment. Obviously, AU refers to RNA, A binding with U. Okay, so those are the rules, as you well know. Why? Well, let's look at the structure of these nitrogenous bases. So first of all, let's have a look. They're hexos, so very similar to hexose sugar, isn't it? But you'll see they're nitrogenous because where we should have carbon, there is nitrogen, and also there's an extra amine. That's going to operate as a base there, isn't it? That's going to operate as a base. That's going to operate as a base. Those of you who know about acids and bases will know what a base is. Okay, that's going to operate base, base, base. Okay, so that's why it's a nitrogenous base. But what we were thinking about was, why does A pair with T and C with G? Well, look, the first reason is if you try to pair A with G, you should note these are called, A and G are called purines because they're double ring. So if you tried to pair those together, you'd get a rather large strand of 4 ring structures, whereas we need to maintain a 3-ring structure all the time because the DNA is like a twisted ladder and all the rungs must be the same length. We cannot have the situation where you can't make a ladder where some are long, some are short, some are long. You can't do that. That's not gonna work. So if you paired these two together, you get a really long rung. That won't work. Okay. However, so what we go for is effectively 3 bases all the time. Okay. But therefore, A could go with C, couldn't it? And then you still get, and G could go with T, but that isn't what happens. A always goes with T, and G always goes with C there. It's worth pointing A and U also go together when we come to RNA. Okay, so what else is going on? Well, let's look at this. The reason is that the way the bases are joined together in the DNA strand is through hydrogen bonding there. And I'll remind you about, as I said to you when we looked at carbs, you cannot have this hydrogen bond here, sorry, this hydrogen atom here cannot form a hydrogen bond. Impossible. Never going to happen. Why? Because it's attached to a carbon. Okay. So such hydrogen atoms do not form hydrogen bonds. So actually you can see the necessity of these nitrogen atoms is that they provide the electronegativity. Chemistry word for you, don't worry about that, biologists. They provide the electronegativity for H here to form a hydrogen bond. If that was attached to a carbon there, you wouldn't get the strong hydrogen bond. And remember, those hydrogen bonds are they're a lot weaker than covalent, but they're a heck of a lot stronger than ID bonds. Okay, we'll discuss that a bit more in a second. Okay, so the reason G goes with C is, okay, first of all, we get 3 bases each time, but also we've got the opportunity here for 3 hydrogen bonds. Okay, and so it's just a question of alignment. The hydrogen on the amine group here has to align with the oxygen there to form a hydrogen bond. Okay, if it was trying to pair up with A, it wouldn't align next to nitrogen or oxygen. Okay, nitrogen or oxygen are capable of forming hydrogen bonds, and fluorine for that matter. That's about it, isn't it? Fluorine, oxygen, nitrogen allow hydrogen bonding. Chlorine, no hydrogen bonding with chlorine, just something you might call a permanent dipole bond. Sorry, that's chemistry, you don't need to know that biology. So this allows 3 bonds, okay, with the when the G and C get together, A and T get together with 2 bonds. Okay, so that's the reason. So we can keep 3 hexose structures each time, and the hydrogen bonds will form. in the correct manner if we stick with A and T and G and C. Okay, so as said before, just like with glucose, we can label the carbon atoms as 1, 2, 3, 4, 5. Okay, and what we find here is when making the sugar phosphate, this is called a sugar phosphate backbone with phosphodiester bonds, we'll find the carbon 3 atom joins with the phosphate. We've said it's condensation at the 3 end and at the 5 end. Now, really and truly, this next bit is no longer on the course. I'm just adding it for interest's sake. What we do for, and also for those who want to go beyond the spec for your essays, one side of the DNA molecule, left side here, joins 3 to 5, carbon 3 to carbon 5. Okay, the other side is actually inverted. Okay, so this is a strand of DNA. It's the ladder structure. It's going to twist, but let's just look at it untwisted, the ladder. So you can see here, carbon 5 joining the phosphate, carbon 3 joining the phosphate. Okay, but if we look here, it's actually carbon 3 joining the phosphate, carbon 5. Okay, so it's upside down. So the left strand runs 5 to 3, the right runs 3 to 5. Okay, so that's just a bit of extra information. The other thing we can see in this diagram, we can see 3 hydrogen bonds there and just 2 hydrogen bonds there, and we can see 1, 2, 3 bases each time. The ladder's got to be straight. Okay, it's a twisted ladder, folks. So why? Why do we have this structure? Now, several things going on here that we need to remember. So first of all, each strand effectively carries a copy of the same gene. So you've almost got you've got the gene on this one strand. You've got a sort of backup copy on this other strand. Okay, if you think about it. So that's one thing. You've got 2 copies of DNA. And so I'm not sure, actually. I don't know if there's a mechanism. You'll understand from your studies before that this would unzip and RNA would come in here. What determines whether, you know, if one side was mutated and the other side not, could it select, oh, that's the mutated side, I'll go to the non-mutated side? That's well beyond the A-level spec. But that'd be a nice, I don't even know myself whether that happens, but you have got 2 copies there. And obviously genetic mutation in the DNA is a serious issue for large organisms like ourselves. So the other thing is having these 2 strands protects the bases and improves molecular stability. Okay, so what we could say is the hydrogen bonds there, think how many hydrogen bonds there are. Here, look, 1, 2, 3, there's loads all down the molecule. And whereas it's possible with an enzyme and not too much energy to break one, collectively, that number of hydrogen bonds is incredibly strong. Okay, so you've got a strong, stable, and what we mean by stability in chemistry is we mean non-reactive. We have a non-reactive, Liam, can you mute your mic, please? I'm buzzing in my ear. Thanks, buddy. So we've got a strong, it's like Theresa May, it's strong and stable. There we go. Anyone remember her and her benighted campaign? Strong and stable, chemically stable, non-reactive. And so the interesting thing about DNA is people, obviously it's abundant in all your cells, and scientists discovered it in about 1860. Okay, so you're thinking 1860, I thought DNA, you know, the age of DNA began with Watson and Crick in the 1950s. Well, that was about determining this structure and function. All the scientists knew DNA existed. They didn't know what it did. It was this non-reactive gunge found in all cells, and no one could work out what it was doing because it was completely non-reactive. So other something like an enzyme in the body, you can say, oh, that's causing a reaction. Okay, this stuff is just lying there, not reacting. What on earth is it doing? Nobody knew. Okay, but you can bet with well, we won't do it today, but when we look at introns and extrons, there's purpose. There appears to be purpose in the design of everything. Everything seems full of purpose. Okay, so that's the strong and stable nature of DNA. Obviously, again, to protect the genetic code, if you like, to prevent changes, to prevent evolution, maybe. 2 strands are required for semi-conservative replication. That's not the strongest argument. So semi-conservative was what I've just been talking about, where these strands unwind and one side gets copied. When you look into the mechanism of that, which we're not going to do today, that's a lot easier to achieve than other methods. Okay, so that's the structure and function of DNA. It is worth pointing out here that we have a fundamental difference between prokaryotes with no nucleus and the eukaryotes have chromosomes and a nucleus. So the prokaryotes do not have, do not have chromosomes. Okay, so what very quickly is a chromosome? A chromosome is formed when DNA winds around histones. What's a histone? Histone is a protein, and it's a bit like having a garden hose. You don't have a garden hose and just throw it in your garage on the floor. It's going to take up too much space. Once you wind it round a circular sort of metal frame, yeah, and you keep your garden hose in a coil, okay, and so that's exactly what a histone does, and we can show that. Here it is. So here's the DNA. We're going to wind round the histone there, and the histones are going to fold up, although obviously this is going to happen when we go into mitosis. Okay, for the purposes of transcription translation, we need it unwound like this. Okay, so in normal non-cell dividing times, there's just going to be long strands of DNA in the nucleus. Once it comes to division time, cell division time, we're going to wrap the DNA around these histones. Histones are going to be folded up, and we're going to end up with this, a chromosome. This is only visible in mitosis or meiosis, folks. Okay, and so DNA wound around a histone is called chromatin. Okay, actually it's saying in interphase. Okay, in interphase it might be there, and in metaphase it will, yeah, be folded up. Okay, but it will need to it's correct what I said, it will need to unfold to do transcription translation. Okay, and so to get a chromosome, we've got to have supercoiled DNA.